Okay, let's begin. So welcome everybody to CTSI seminar for this week. Today we have the pleasure of having Dr. Catherine Wu speak today from the Dana-Farber Cancer Institute in Boston. I know I had some programming notes. So this is CTSI seminar, but it's sponsored by both the I3T and the Cancer Center. So it's a joint thing for our CTSI seminar series. And just programming notes, we have several new I3T talks coming up next week. There is a research in progress with trainees from the Bensinger lab and Manish Bhutt's lab. Following, we have Amanda Lund from NYU talking on the 28th. So again, it's great to be able to introduce Dr. Kathy Wu today. Now, we certainly see each other at the Parker Institute meetings all the time. The next one's going to be nice. Yes. Dr. Wu did her undergrad training at Harvard. She majored in biochemistry, followed by medical school training at Stanford. And then she has moved back to Boston to complete her residency and fellowship training, correct? At the Brigham and at the Dana-Farber. And since 2006, she has put up on faculty and risen through the ranks all the way up to where she is at the moment. She's currently chief in the division of stem cell transplant and cellular therapies at the Dana-Farber, faculty advisor for the Dana-Farber Translational Immunogenomics, and member of the Broad, and program leader in the Human Biology and Translational Medicine, one of the academic grad programs there. She has too many words to talk about if we want to actually let her speak, but she's a member of the American Society of Clinical Investigation, elected member of the National Academy of Medicine, and principal investigator on many PO1s and a SPORE project, multiple extramural grants at Harvard. And I think what you will see today is she has devoted a lot of her time to looking at the targets of the immune response and different types of cancer. I think she focused on chronic lymphocytic leukemia, I think, to begin, and now she's branching out into multiple types of solid tumors. She's looking at neoantigens, multiple high impact publications that you will get to hear about today. She's spanned the course from preclinical to clinical. She holds the IND for Neovax, which is this neoantigen vaccine. She's a great example for the trainees of a physician scientist who sits right at this boundary between what is preclinical and clinical, starting new trials, taking what's done in the lab to new trials in patients. She's a great example of how this can be done. So today, the talk is entitled T-cell specificity, phenotype, and dynamics, the impact on cancer immunotherapy. Please give her a warm welcome.
Okay. Well, so thanks for that very, very warm welcome. These are my disclosures, and I have to say, you know, it's California has always held like a very deep place in my heart. It's where a lot of my family is. It's where I met my husband. It's formative years in medical school. And I did start my day this morning in San Francisco. And this was a couple of days ago, kind of flying down into SFO. And certainly when you see that view, there always are heartstrings that kind of pull at you. So green. I learned a new vocabulary word that I never heard before, atmospheric river. That was like a new one. Like we've been dealing with Arctic blasts, polar vortexes. So this was a new one. I think at some point yesterday there was, I think, mist coming horizontally at us, not vertically. So that was a new experience. This was this morning. So a beautiful sunrise out of SFO. And it is raining here, but I have to tell you, we did have a snow day yesterday. So that's what's going on. And that's Boston right now. This is actually Arlington, actually out in Lexington. And so I think that our plan was always to come back to California, but life takes its course and we've continued on in Boston. So anyway, delighted to be here. And as introduced, we're going to talk about T cells today. I think we all recognize that T cells have wonderful capabilities, many, many specific capabilities that have made them of great interest to harness when we start to think about immunotherapy. And I think this audience knows very well that at this day and age, we have a lot of different flavors of immunotherapy that are out there that are all kind of capitalizing on specific aspects of T cell function. They are not all the same. They act on different aspects. And it is a very, very exciting time for cancer immunotherapy and cancer therapeutics in general, because although immunotherapy has long been envisioned as really the magic bullet, the way that the path toward eradicating cancer, it's really not until these last 10 years that we've really seen everything come to fruition. Despite that, we know that there are lots of challenges that still face us. Cancer is still a very, very tough problem, not the least being that we're dealing with a heterogeneous entity. There's a lot of genomic heterogeneity within cancers. We know that cancer evolution is happening. And at the same time, we also know that it is co-evolving together with the host immune response. And this was kind of a figure that we kind of put together a number of years ago, is a little way to kind of, in our mind, redraw what Bob Schreiber had already talked about, the 3 E's, and kind of what is happening over time. Unfortunately, for most of us, we are diagnosing cancer at the very end of the story, where the cancer has already evolved, has already put in place all sorts of different mechanisms to kind of inactivate a functioning immune response. And we're left with asking, how is our immune system functioning to control the cancer? Are the right cells at the right place at the right time? What is their functionality? For example, if we're going to use immune checkpoint blockade, is there going to be a response or not? Because did the right cells with the right function get there? And then we also have to ask, well, what is the role of tumor antigen in shaping these T cell responses? And so my group has really been interested in this kind of co-evolving question and how to address these challenges. So what I'm going to go over with you today is some of our recent work. I've really tried to focus on unpublished or recent work. And the first part I'm going to talk about is what is the native landscape of the tumor reactive tissue infiltrating T cells that are there. And this has really been work that has been spearheaded by a talented instructor in my group, Giacomo Oliveira, and in another story of pathologist scientist Brian Yorgulescu. So I think when we talk about tumor-specific T cells, tumor-specific TCRs, we do have a very fundamental challenge, which actually they're rare cells. So when you kind of look at the tumor microenvironment in bulk, most of the cells there have been recognized to be bystander cells that actually don't have specificity for the tumor. So it's actually kind of hard to pick out those specific T cells that we want to study. And yet, if we could, then we could start to understand the phenotype and the specificity and all that good stuff. So our approach in general in our group has been to leverage all the fantastic single cell technology that we now have available and really focus first. If I do this, will the online people see this, or should I be using, maybe I'll do this. Yeah. So I think that our approach has been first to think about the phenotype. So with single cell transcriptome sequencing, we now can focus on immune cell populations that are infiltrating, get a sense of kind of their transcriptional program. And then single cell technologies can be easily also approached to look at the T cell repertoire. So through single cell TCR sequencing, we gain information about the CDR3 alpha beta chain sequences that define a TCR and are molecular barcodes of a particular clone. And the third aspect, which we've really been interested in, is what are those T cells seeing? So what is the antigen that is in contact with the TCR? And here we've been able to use a number of different approaches. We've long partnered together with our proteomics colleagues to look directly what is presented peptides on those tumor cells. We've also devised kind of predictive algorithms, and in one actually nice marriage, I'm really not going to go over it too much, only to say that we were able to generate these monoallelic cell lines for 95 different individual HLA alleles, and then directly elute off peptides that gave us 180,000 peptides from which we could train our predictive algorithms across different alleles for a class 1 epitope binding. And then there are also commercial sources of peptides, for example, against viral antigens that can serve as a foil. So this has kind of been like the tools that we've been using to try to approach this problem. And Giacomo, when he first joined the lab, we were already moving in the direction of performing TCR reconstruction cloning, but it was super slow. And so one of the first things he did as a postdoc was try to come up with at least a medium throughput approach. And this was really to take that CDR3 alpha beta chain information and then miniaturize and parallelize a cloning strategy that does use Gibson assembly. And then once those TCRs are reconstructed, then to introduce them into T cells. And then he came up with this very clever combinatorial dye labeling approach so that each of those individual clones have a different color. You can kind of make the mixes. So in one tube, you can at the same time interrogate multiples of these individual clones. And the first question he asks is there or is there not tumor specificity? So that's the first gate. For we first focused on our studies, as I'll show you, in melanoma, where we had available cells from which we could generate a patient-derived cell line, so he could ask that question. And then after that, once you establish which TCRs are tumor-specific, what is their antigen specificity? And here again, we used our different tools to come up with targeted peptides that we could pulse on top of autologous EBV immortalized B cell lines. And the readout we used was CD137 or 41BB by flow cytometry. And in this example, you could see that there's reactivity against melanoma, interferon gamma stimulated melanoma, but not against autologous PBMC's B cells, nor the EBV immortalized B cell line. That was our general approach. And through the process, we learned a lot of interesting things. Again, this is published work, but I just want to give you some of the highlights. In the CD8 TILs, we came up with a UMAP that we called the turtle, because that's what it looks like. And what we could see is that there were, in general, two big phenotypic clusters. So one was the reds and the oranges and the yellows that transcriptionally look like exhausted T cells. And then here in the tail of the turtle was the non-exhausted memory T cells, so in the blues, and the clonally expanded T cells, and we could tell that because of the single cell TCR sequencing, were here in the head and the shoulders. And what Giacomo did was he actually focused on the dominant clonally expanded TCRs that were coming from these two different regions, cloned them, expressed them, tested them against melanoma, and turns out that actually the majority of the tumor reactivity is in this red zone, and they were either melanoma associated or neoantigen specific. They were in this area, whereas what was emerged as the non-exhausted memory T cells, they were only occasionally tumor specific. And this is where the viral specificity was, because we had paired PBMC B cells and EBV immortalized B cells, and we could see that actually the reactivity could be ascribed to the EBV reactivity. It turns out on the CD4 side, we also see something very similar. So it's the exhausted TCRs coming from the exhausted T cells where we see the tumor specificity. We see rare tumor specificity in the non-exhausted memory. But then what was super interesting is that we saw a very large Treg population, and it turns out that this was also mostly tumor specific and actually specific against neoantigens. Now, these 4 patients that we studied intensely, this is just a pie chart of kind of as we started to deconvolve what was the antigen specificity. And I think what's striking is that the antigen specificity landscape of these 4 patients are really quite different. And it really tells you that this is a very sort of heterogeneous kind of immune response. Patient A and B had more reactivity to the melanoma-associated antigens, where patient C and D had more reactivity that we could deconvolve that was against the neoantigens. And I think this already raises the question of what are the tumor intrinsic features that are driving this type of antigen response. And just to reiterate again, those specificities, whether melanoma-associated or neoantigen reactive, are in the head and the shoulders, whereas the viral specificities are really here at the tail. And despite the melanoma-associated antigens and neoantigens being in the same phenotypic region, there are definitely differences between those TCRs because, for example, at the level of TCR avidity, we see that the neoantigen-specific TCRs are far more with high avidity than the melanoma-associated. So we've started to now expand beyond melanoma because just because we see it in melanoma, melanoma does have a very dear place in our heart, but it may or may not be true in other tumor settings or contexts. And we've had available to us data sets in renal cell carcinoma and head and neck squamous cell carcinoma. And I think you can appreciate that, again, we're seeing these red and blue zones. And from the melanoma data, we had constructed a signature of tumor-specific reactivity as well as for viral specificity. And when we kind of superimpose that signature in RCC and head and neck, we see that there again is a close apposition of that signature to where the phenotypically exhausted T cells are. So I think some of the same themes are going to come through across the other tumors, and I'll go into that a little bit more. Another dimension that we've been trying to look at the baseline response. Again, I think it's instructive to look at the baseline response because as we're kind of going into our therapies, we want to know what is the hostile environment that we're dealing with. We've been able to partner really closely together with Fei Chen to ask the question, how are these clonotypes organized in space within the tissue? And so Fei has created this very wonderful and informative spatial transcriptomics platform called SlideSeq. And really the concept there is in the same way that single cell, for example, 10x technologies have allowed us to take suspension cells and connect with beads and to perform a single cell analysis with those type of workflows. This is a rather similar workflow, except the beads are now organized as what we call a puck. And each bead has kind of XY coordinate information. So after they kind of go through their cDNA library transcriptome processing, there is computational reconstruction with that knowledge of the XY coordinates. So for example, you can reconstruct in a tissue, and I'm summarizing a lot of published work that Fei has already generated, where you can look at different cellular structures in tissue space. He's actually also reported out an approach to use this technology adapted to looking at DNA alterations. So for example, you can look at copy number changes. And our role in this project was to see if we could bring in TCR, because we had already generated an approach to do targeted TCR detection at the single cell level in a plate-based manner. And it turns out that those protocols kind of merged together really nicely. We've already reported out the ability to do this, and it's actually been very fun to kind of go through some of our initial tissue samples. This was an example of a renal cell carcinoma pulmonary MET. And what was special about this particular piece of tissue is that it just works out that there was the MET, and it was just juxtaposed against normal lungs. So there was a kind of a tumor normal interface. So some of the analyses that we were able to do was to examine to what extent clonotypes that were expanded, were they actually enriched within the tumor, or were they actually depleted or excluded from the tumor? And I think you can appreciate that, in fact, we did see a number of different clonotypes that were in the different points of space. In another example of tissue that we looked again at melanoma. And in this example, we were able to look at different regions of interest. And both in the RCC sample, in the melanoma, we were able to appreciate that there were tertiary lymphoid structures. And you can see that based on the transcripts, we could discern that they were in the blue B cells and the orange CD4 cells. We could look at the clonality. We could see that it was a polyclonal mix of CD4 cells. And we could overlay, and this was again through the TCR sequencing that we could begin to look at those kind of clonality measurements. And finally, going back to the sample in a very super interesting finding that we reported out, this melanoma sample actually had two lobes. So here, kind of by the masking, you can see there was a blue lobe 1 and orange lobe 2. And again, when we started to look at the clonotypes, we noticed that one particular clonotype was actually preferentially in lobe 2 rather than lobe 1. That's interesting. So let's start to look, can we do some sort of neighborhood analysis to see what are the other cellular players in that region? So it turns out that as Sophia Liu, who really led the study, started to look at that particular T cell clonotype compared to surrounding monocytes, what emerged as a very dominant transcript was CXCL10, which is a chemokine and an attractant for T cells. And she also did an analysis to look at what is the neighborhood of the tumor cells around that T cells. And here there's a negative association with MGST1, which has been actually associated with immunotherapy response. So I think this just gives you a flavor of the sort of analyses that we're beginning to embark on. And we're in the midst of actually trying to collect a larger number of tumors so that we can do this a little bit more at scale and trying to bring all the different layers together. And so just to summarize this part of my talk, we're at a time where we have really exciting tools that are allowing us to directly look at human biospecimens and kind of learn their secrets, I suppose. And I think that when I think about the TIL analysis that we've been doing in the melanoma, it's a little bit like tumor immune cell archaeology. I think we're seeing traces of past tumor immune cell interactions that happened. We see kind of the effects of that interaction, namely the exhaustion that we see for those T cells that are exquisitely tumor reactive. A point of emphasis is that the TCRs with the most potent tumor specific recognition at baseline have this exhausted phenotype. So I think that's an important takeaway. Those specificities can be directed against really a mix of different antigens. And as much as some of you may know that I am a proponent of neoantigens, I am also at some level agnostic. So I'll go with whatever works. And there's definitely melanoma associated T cells, T cells that recognize melanoma associated antigens. And we've done functional studies to show that they recognize the tumor very potently. And I would also say that the spatial organization is, I think, we don't have data yet, but we're in the process of working through that. I think that's going to give us another dimension to help us think about what are the kind of T cells that perhaps might be the most important or play a prominent role in this kind of anti-tumor response. And it's a question mark in our mind, but is it somehow related to the intrinsic features of the tumor? On the CD4 side, I didn't want to burden us with too much past stories that have already been published, but I just want to say that through looking at the CD4 TILs, I do believe that we uncovered a subtype of melanoma because at least some of the cases that we looked at were unexpectedly high for HLA class 2 expression. And our studies do support this notion that there's direct engagement of immunosuppressive Tregs as a mechanism of immune evasion for that subtype of melanoma. It's crazy. So I think just to summarize this part of the talk, I think, again, the dominant message is that at some level, the T cells we want the most are super tired. They're exhausted. So this leads us to ask the next question, which is, with therapy, can they be revived or can they be replaced? So the other part of my talk is thinking about the dynamics of T cells in relationship to therapy. I'm actually going to talk about checkpoint blockade and vaccines. I thought for the sake of time, I'd leave out transplant, but I'm happy to talk to you guys about transplant anytime. So I have a little bit of blood malignancies in here, but not related to transplant. And again, this is work that has really been spearheaded by super fabulous trainees in my group. The first story I'm going to tell you is related to the new exciting area of neoadjuvant therapy. And I think that this is the idea that rather than go straight to surgery, that you might provide some sort of therapy in advance that will lead to tumor reduction and hopefully lead to better outcomes. And there's a lot of promising studies in this area. This is a study that we worked together with Ravi Upaluri, who runs the head and neck program over at the Brigham in conjunction with Dana-Farber. And he had already designed a few neoadjuvant studies where patients were enrolled and they received pembrolizumab, so anti-PD-1, in advance of surgery. And from an immunobiology perspective, the beauty of this is that we actually have access to pre-treatment tumor specimens, and then defined by a clinical trial, a post-therapy specimen. So it gives you a window to be able to see what was directly the impact of the said therapy. Now, what Ravi had already seen, he had already published a trial looking at the first cohort of the study, in which patients got 1 dose of pembro. And now we partnered together as he was gathering data together on his second cohort. And he had already been able to see, if you focus on the red, that with the 2 doses of pembro, patients seem to have better pathologic responses. So looking under the microscope, looking at the degree where there was evidence of tumor regression, the second cohort seemed to do better. But I think the important thing from, again, the immunobiology perspective is that there was good sample collection. There was a very tight window. So all of those patients, again, for a human study, we're looking for opportunities to have a homogeneous cohort where there's not variability all over the place. So I think that was great. And the median interval between those 2 time points were 35 days. So I think that's also relevant, but again, very tight. And so we focused both on the question of baseline samples, but then for another subset, we had responders and non-responders that were pre and post. And we used our methodology du jour, which was focusing on T cells that are the TILs, using single cell RNA sequencing and then TCR sequencing. Now at the baseline, I already showed this UMAP image. We defined the different sort of T cell clusters that were the TILs that were in the tissue specimens. And it turns out that the population that was the most distinct and different between the annotated responders and non-responders were this exhausted population. So that's shown here. And really, we didn't see much dynamic differences between the other populations. And even more finely, because you'll see that it's not a monolithic single red color, but in fact, there's different clusters here. There were differences among these exhausted populations. There was one population in particular that seemed to be different between responders and non-responders. And this is this exhausted population that was characterized as having high cytolytic potential and a tissue resident memory program and a lot of granzyme in there. There's also this transcription factor CNF683. Whereas the non-responders, what was more enriched in the baseline for non-responders were these truly highly exhausted T cells that had high expression not only of TOX, but also TIGIT, PD-1, CTLA-4. What about pre and post? If we look at the 2 time points, the most dynamic population, again, was this exhausted population in the responders. We see actually this exhausted population appear to contract. And we really didn't see that in the non-responders. And again, it was more this kind of high cytotoxic potential population rather than this, and the non-responders by contrast seem to have, if anything, some expansion of the really exhausted population. So what is the mechanism by which this is happening? Others have, including the group of Howard Chan, have looked in basal cell carcinoma, and they've actually talked about clonal replacement as something that happens with PD-1 therapy. And we looked at the clonotypes, we looked at that pre and post. It didn't really seem to be clonal replacement. In fact, what we saw mostly were the same persisting clonotypes pre and post, so that's shown here, and even loss of particular clonotypes. So we're not seeing clonal replacement. We're also not seeing clonal revival. So this is another mechanism that Ziming Zhang has put forth. When we look at the phenotypes, the signatures of those expanded clonotypes pre and post, there's really no difference over time. So what do we think is going on? We'd like to propose that there's probably what we think is cytotoxic revival. So because the cells that are the most dynamic and the ones that are contracting are those that have high cytolytic potential that are ZNF 683. This population is contracting at the same time that the tumor is shrinking. So we actually think that the contraction is related to antigen clearance. So this population is then not driven to expand any further. And so I think trying to put all of this kind of picture together, we do think that at least in some cancers like head and neck, we do see already differences in the pre-treatment moment where non-responders have really truly very exhausted cells, whereas responders have exhausted T cells, but different flavors of exhausted. So a little bit more of a heterogeneous population. And that in the post-treatment setting, what we see is that the responders are actually going through the cytolytic revival. There's actually cytotoxic activity that we're inferring, of course, and that's leading to tissue regression and then antigen clearance. We also don't exclude that these other mechanisms may also be at play, because I just want to emphasize that our windows of sight are those first 35 days. And this could be something that's happening very early on, whereas the other studies have looked at later time points, and there may be sort of cascades of response that are happening in time. But I think this does give us maybe insight into maybe ways that we can already understand, even in advance of therapies, who's destined to respond or not respond. I think another area that, we were always looking for orthogonal ways that kind of reinforce the bits of truth that we are hoping to look for. And I think one thing that's been super interesting in our own group, it was mentioned that our group works on many different cancers, and Erin Perry is a very talented instructor, and her area of interest is B-cell lymphomas. And what Erin's been actually super interested in is Richter syndrome. So this idea of histologic transformation. So how is it that you can have an indolent small B-cell lymphoma, and a minority of those patients have the misfortune of then transforming to a histologically large, angry, diffuse large B-cell like histology? What are the cellular programs that are driving that sort of change? And this is also particularly striking because at a time where there is a lot of therapeutic opportunities for chronic lymphocytic leukemia between the targeted inhibitors and whether it's BCL2 inhibition or BTK inhibition, there is very little we can still do for patients with Richter syndrome. These are the patients we tell them to put their affairs in order and to spend time with their families. And I think, just as an example, Erin had collected a series of more than 50 patients where we had paired CLL and Richter's. And I think the point I want to emphasize in terms of this swimmer's plot is that all the black is all the different samples we collected during the green, the CLL phase, and the yellow is the time of transition to Richter's. You'll notice a lot of red crosses. These patients did not live long. And again, what I want to emphasize is that the CLL, the scale is in years, whereas on the Richter side, it's in months. So I think this just gives you some kind of concept of how different these different entities are. Now, Richter's, again, for the blood malignancies, we've often had the advantage and the luxury of being able to easily access tumor from the blood. And this has led us to do lots of studies in chronic lymphocytic leukemia. Richter's DLBCL is more like a solid tumor. It's hard to get access to those tumors. I think that has been a challenge in the field in terms of studying this entity more deeply. But what I will say is that in the last, I would say 2 or 3 years, there have been a lot of very cool studies, both on the human side and on the murine model side, that have been trying to integrate all these different levels of genomic information so that we can actually do better in terms of our understanding of the biology. And just one slide on some very beautiful work that Erin did. She was, because of this very nice set of samples where there were paired CLL going to Richter's, she was able to identify the driver mutations, also the cellular pathways associated with response, histologic transformation. She could actually identify that there were molecular subtypes of Richter's that are defined by different combinations of mutations. through single cell analysis, she could see that she could actually identify those subpopulations in transition from one histology to the other. And ultimately, she was able to use the genomic information to help us get to non-invasive early disease detection. So there are some cases in which we're able to find those copy number changes in small amounts of cell-free DNA, even 7 months before the actual diagnosis of Richter's, which I think opens therapeutic opportunities for those patients. But that being said, so, Richter's is a problem, but one of the surprising things that has emerged in kind of the therapeutic landscape for Richter's is that actually about 50% of patients do appear to respond to PD-1 blockade. This is unusual because, there's been it's kind of like the highs and the lows, Hodgkin disease, wow, great responses. And then you get to myeloma, CLL, the myeloid leukemia, it's kind of not been great. So the fact that we actually saw across a number of different clinical trials over the last couple of years that there's 50% of responses makes us wonder. And so we had the opportunity to work very closely with Nitin Jain and Bill Weirda and MD Anderson, who had just recently reported out their trial. They had responders, they had non-responders, they did dedicated marrow specimen biocollection. This was a group of patients that received Nivo first, and then they got ibrutinib. And there was kind of response assessment at these defined periods of time. So Aaron, together with Nitin, was able to identify a small discovery cohort of 4 responders, 2 non-responders, a few CLLs, and perform a single cell analysis. And the analysis was really quite interesting. So as we put together all of these different cells across the different patients, annotated for whether or not the cells came from a responder or a non-responder, you'll see a concentration of purple here in this region. And that also seemed to be where this transcription factor, which I already mentioned before, ZNF683, appeared to also concentrate. And in terms of kind of gene set enrichment analysis, the top ZNF683 was a top regulated transcription factor in the responders, whereas for the non-responders, it was tox. And then as we started to perform trajectory analysis, it turns out, maybe this is a little bit reminiscent of what we saw in the head and neck cancer. There is one trajectory that is going in the direction of like deeply exhausted cells, but there's another trajectory where these are kind of maybe intermediate exhausted. They're effector memory CD8 cells that are ZNF683 high. And on kind of further bone marrow. So all of this was bone marrow cells, and on a little bit more inspection, it turns out that when we looked at a meta-analysis performed by Ziming Zhang, they had also defined in the solid tumors this ZNF683 population that matched very well with what we saw. This ZNF683 population was in cluster 1. So when we looked at a melanoma data set, whether it was either for ZNF683 high or using in other analyses, this is cluster 1 or the 1 transcript, we actually see it separates out kind of survival responses. Aaron did perform some very nice studies to overexpress ZNF683, and for the sake of time, I won't show you, but it does implicate ZNF683 in the regulation of key genes in T cell differentiation and activation. She also used cut and tag to start to look at this. And just again, kind of a little bit reminiscent thematically to some of the things that we saw in the head and neck situation, these 2 plots are organized. So this was the clonotype information from a responder on the left, a non-responder on the right. The bar graphs are organized in terms of the most expanded clonotypes. There's a difference in the scale on the y-axis, so much more expanded in the responder than the non-responder. And the red is this cluster 1 that is ZNF683 high. And I think the point that I want to emphasize is, again, in terms of the phenotype, the transcriptional signatures of those different clonotypes, they're really not changing pre and post. Again, there's this idea that there's kind of already a pre-existing response that the therapy is helping to play out. And again, there's perhaps these were somewhat different time point than what Giacomo had looked in the head and neck cancer. I don't think that we necessarily see contraction, but we do see differences between the responder and non-responder. And so I think what I've kind of shown you is like, I think we think about the therapies that we're giving, and we're also thinking about, in the orange, are we actually acting on a pre-existing anti-tumor T cell population, or in the green, is our therapy actually, sorry, I'm going forward, is our therapy actually acting on bringing in new T cells onto the scene? And so I think with the checkpoint antibodies like anti-PD-1, I think we're really acting and reinvigorating, expanding T cells that are already in the TIL landscape. I think that's what I've tried to show you, both in through kind of clone tracking, both in the marrow infiltrating setting, as well as in a tumor like head and neck cancer. But I think that we do have an opportunity with an alternatives type of therapy, such as vaccines, where we can not only amplify pre-existing responses, but also elicit new responses. So I'm going to pivot and talk to you more about our work in vaccines. And I think this is really, vaccines have had a pretty long, perhaps checkered history. It's not a new concept. They're complicated in the sense that there are many components to it. But our group at least perceives vaccines as maybe a bridge over troubled waters, shall we say, presented to us by the cancer. And if formulated so that the vaccine addresses multiple different epitopes, maybe this is an approach that can directly address the challenge of cancer heterogeneity. And certainly, as I'll start to talk to you about, perhaps these are promising in combination with agents such as checkpoint blockade, knowing, of course, there are still, even to this day, many unknowns about vaccinology. But one of our hopes as we started to get into the space and working with cancer neoantigens is that maybe we could at least stick that feature while iterating around the others. And this is just a older visual that we generated close to a decade ago, where we started to, we were trying to visualize and articulate this notion that's been in the air for decades, which is that tumor neoantigens have been long conceptualized as ideal tumor antigens. Why? Because they have, they're born of somatic mutations that are part and parcel of cancers, and so highly exquisite in tumor-specific expression. And then akin to pathogens, because they're somatic events, there's a lack of central tolerance, so the capacity for high immunogenicity. And this may be a different place than where we've been thinking about shared overexpressed antigens that are variable in terms of tumor specific expression and variable in terms of their immunogenicity. But I think fast forward to where we are now, I think, what's really changed for us over the past decade is genomics and the ability on in a cost-effective manner to scan individual tumors, identify mutations, and then begin to put it together with tools that allow us to perform prediction for class 1 binding, depending on the personal combination of HLA alleles of that tumor, and get us to target antigens that we can go after. So this is the concept that we've had for a while. As I mentioned before, we've iterated to try to improve the algorithms by incorporating mass spec data, and there's still much more room to improve. But this has been since incorporated in a number of different clinical trials that this audience may be aware of. But just again, to run you through it, as we were getting into our genomic revolution, together with Nir HaKohen and Ed Fritsch, we began to conceptualize, couldn't we readily procure tumor from patients? Couldn't we go through this sort of genomic analysis, come up with a manufacture strategy, and then get it in a syringe together with a strong adjuvant? And at the time, you have to look at the date that we generated this figure, we went with synthetic long peptides by virtue of the fact that there was a long history of safety data in peptides in patients. Craig Slinghoff had already reported by then combining 15 different peptides against melanoma-associated antigens, and this was safe. There wasn't adverse events like autoimmunity. And then also Case Maleev at that point had already reported both preclinical and clinical data using peptides of 20, 30 mers, not the 8 to 10 mers that we typically think of for class 1 T-cell responses that would require and necessitate a professional antigen presenting cell to process that long peptide and harness both CD4 and CD8 sort of responses and create a robust response. So as we started to think about a therapeutic strategy, we did go with synthetic long peptides. We put this together with best-in-class adjuvant poly-ICLC. We wanted to target up to 20 neoantigens to some fudge factors to generate a multiple polyclonal response. And we also tried to put our peptides in separate pools to decrease antigenic competition. And again, just to summarize published data, I don't think it's a surprise. Our studies are here in the red. I don't think it's a surprise that maybe the first setting that we went after this approach was in the adjuvant setting for high-risk melanoma, a very tried and well-traveled path for novel immunotherapeutics. I also think good ideas never happen in a vacuum. So our study was co-published together with a group of Uber Sahin. So they took a very similar approach, except that they used intranodal injection of a RNA vaccine, which was a very new concept at the time. Fast forward now to where we are at the heels of our pandemic and the knowledge that we have now. But back then that was very new. And this was reported 2 years after Beatrice Carreno and Jerry Lynette had also used the computational approach to predict personal neoantigens that were then pulsed on dendritic cells and given to patients with melanoma. 2 years later, we reported a newly diagnosed GBM, a very challenging setting, an area of great clinical need. And I'll show you a little bit of that data. But I think in aggregate, I think all of these studies do tell us safe, feasible as an approach, highly immunogenic, and indeed across different groups promising anecdotal combinations with checkpoint blockade. Perhaps the most one of the more exciting results from our first pilot study were these 2 patients that had stage 4 melanoma who did receive in the blue our vaccine. The schedule was 5 doses in the first 3 weeks and 2 boosting doses thereafter to try to mimic what we think is happening in a viral infection. So high antigenic exposure and then induction of memory. And it was without surprise that they both suffered recurrences thereafter, but then their treating physicians gave them anti-PD-1 therapy. And what was impressive is that they both went into complete clinical response, and this has now endured more than 4 or 5 years later. This is some of the early radiographic data that we saw where, these yellow lesions arrowed here then went away. And so I think that one of the fun things about having a investigator-initiated study is that we actually keep close tabs on our patients. We're able to follow them closely. Our early work was really focused on looking at the immune responses after the second boost. But we were then able to look 3 or 4 years later. So what's in the dark gray was the intervening time since our first report, and the patients very generously and willingly provided us with extra tubes of blood that we could then analyze. And the results were really pretty interesting. So the first question we asked is, are we generating a memory response? We sure do hope so. We were able to partner together with Kai Wu-Furfenig, who helped us construct class 2 tetramers so that we could directly pull out from the blood neoantigen-specific T cells. And then we followed those tetramer pulled out cells over time and did this for a few different patients. And I hope what you can appreciate is that then we performed single cell transcriptomes. We could find distinct clusters. And it looks like when you look at it in a time resolved fashion, there is in fact a cascade that we could follow that these neoantigen specific T cells were following, which is going from a naive signature to that of cytotoxicity, then immune contraction, and ultimately generation of memory. That's good. Well, what about the clonotype diversity? Those neoantigen specific T cells did appear to not only diversify, but also persist. So In this example, this pie chart, each color is the first time that we see a clonotype. And I think you can appreciate that the red persists across these many, many weeks, but there is also introduction of more colors. And it's telling you that there are many, many new clonotypes that are kind of expanding against the same neoantigen. And functionally by ELISpot, so this is a assay in which we're detecting gamma secretion when peptide and T cells are coming together. Even 4 years later, in the blood of these patients, we can still see strong immune responses to the original immunogen that was in the vaccine. So that was very cool. And then finally, we looked at epitope spreading. Now, remember, these were patients who were in the adjuvant setting. They had surgery. We did not have tumor from which we could follow whether or not there was a direct anti-tumor response. And so our approach was to ask, was there any evidence of on-target tumor killing, for example, by epitope spreading, which means that there are responses against tumor epitopes that were not in the original vaccine? Maybe by the killing of the tumor, there was access of antigen-presenting cells to additional epitopes that could then stimulate secondary responses. And the answer is yes. We were able to find epitope spreading both against mutated and melanoma-associated antigens. And Neon Therapeutics had performed a separate study in melanoma and was able to show in a larger series of patients that if they could demonstrate epitope spreading, that those patients had a better clinical response than those that did not have epitope spreading. I think another piece of data that has been super interesting in terms of thinking about what our vaccines might be doing is actually we've tried to use the TCR information to help us track whether the T cells that we're stimulating are actually getting to the site of tumor, because there's a surfeit of evidence that suggests that we need the tumors to actually get in there. One of the first studies that we tried to use this approach was actually in GBM. And GBM is, as you may very well know, a very difficult tumor to study. I would say that most surgeries that are happening for GBM, I like to say, either happen after 11 p.m. or during the weekends. So it's actually super hard to procure tissue to do the things that we like to do in the lab. But lucky patient 7, for some reason, all the stars sort of aligned. And this was a patient where at the time of recurrence, we were after vaccine, we were able to collect tissue, sort out those T cells, and perform transcriptome sequencing. So this is an example where in this analysis of patient 7, every single dot is a T cell, and if they're grouped together, this is like a specific clonotype. And then this particular patient, we had already been able in the peripheral blood to analyze what was the dominant immunogen that was stimulating a neoantigen-specific response. So we knew the clonotypes of those neoantigen-specific T cells. And lo and behold, there was actually overlap in what we kind of stimulated in the peripheral blood with what was infiltrated in the GBM tumor itself. And then upon reconstruction and cloning, and interrogating with that immunogen, we were able to confirm that some of those overlapping TCRs were indeed against the mutated epitope that was part of the vaccine and not against the wild type. So this was, I think, our first foray into kind of really using TCR barcodes as a way to do that type of tracking. In another example, in metastatic melanoma, where I already described a patient who had gotten the vaccine, recurred, then got the anti-PD-1. We had performed the single cell TCR sequencing at these 2 different time points. And what these kind of overlapping green and orange circles are telling you is that, yes, persistence, there are lots of overlap in terms of those neoantigen-specific T cells that are persisting in the blood over time. And that's the ones that we know because of their single cell TCR sequences. We know that our neoantigen specific are here in the red. What was interesting is now that we knew that information, we use that again as a molecular barcode to look at the recurrent specimen. And when we did bulk TCR sequencing, we could match those barcodes and in fact saw evidence of those neoantigen specific T cells at the site of the recurrence. And one might speculate that maybe this patient responded so quickly to the pembro because the vaccine was actually able to get the right soldiers at the site of tumor. So when the pembro was given, things could happen. So So it's an exciting time. I would just say that, this is close to some of my final slides, but I just would say the winds are blowing in the right direction. Vaccines have had a long checkered past, but I do think that right now, through the accumulation of different pieces of data that I've started to show you, I think Ugur was also able to show decreased recurrences after RNA vaccine with melanoma. Some new data from Vinod Balachandran that he reported out in ASCO is again using an mRNA vaccine in pancreatic cancer, tough disease. They're not seeing any recurrences and they're going out 2 or 3 years. And that's actually pretty spectacular. Both Vinod and our group have been using, again, clonotypes to track T cells to the site of tumor. And many of you will know that there's been a press release of probably a positive signal between the Moderna-Merck partnership on a vaccine for melanoma. And so I haven't seen any of the data. I don't know anything about it. The press release is out there. We're waiting with bated breath, but I think it is a very exciting time. So I think this is a good time to ask, is this the moment for vaccines? Or, there's so many things we have to work on to try to really make our vaccines the best possible, but there certainly are a lot of encouraging signals. And I think this gets us to this question of how can we use the lessons of the pandemic to help us get to scale so that we can really see some clinical signals. And I think this gets to the issues of cost and time, which are not really what an academic group is well suited to do. Well, I think we can supply the signals to help, but it can help hopefully create partnerships that can actually get us in the right direction. And so that's all I have to say to you, that I think we're in an exciting time. I think we can learn a lot from T-cell dynamics in a lot of different settings, as well as in baseline. Maybe it can even help us predict responses to our therapies. I think tumor antigen is super important in terms of maintaining T-cell populations at the tumor milieu. We're super excited about the ZNF683 signal that we've been finding. We're poised to really go more deeply into the biology. And then in terms of vaccines, we've been thinking about epitope spreading. Is that a good signal for us to think about how immune signals and surveillance is maintained? And then we're always looking to see from our clinical trials of what we're learning. So I thank you. This is really a partnership between our groups at the Dana-Farber, where we have very nice translational infrastructure using different immunobiology assays, but also clinical operations, and really the ability to run these kind of investigator-initiated clinical trials. We work very closely with our friends at the Broad 3 miles away across the river, near Cohen's lab, but also Steve Carr's group on the proteomic core, Gadi Getz, and also the genomic platform, which provides us with the CLIA sequencing. And we've actually also worked very closely with many different basic labs at the Broad at MIT. And this is just a fun image that we shot that Immunity had their 25th anniversary and they asked a lot of immunology labs to provide snapshots of their lab for their cover art or something like that. So we did this right after lab meeting. This was kind of some of the different tumors that we work on, and lots of different folks that are working on harnessing T cells populations to go at the tumors. And I thank you for your attention.
Okay. I get to ask first question. So Rafi Ahmed was actually here last week, and he was talking about, again, this progenitor exhaustion, or he calls it the stem, like numbers people were talking about. And he was saying it's moving into the neck, looking at that, kind of parlaying it back to his LCMV. And he was saying, like, the reservoir of these cells that expand, the checkpoint locator, this is population, and you can see them in the tertiary lymphoid structure there. I wonder if you're starting to connect.
Yeah, I mean, I think that's the beauty of all the different tools we have right now, which is that we can start to connect those pieces. So kind of trying to move from a descriptive aspect to really bring on. So we haven't looked, but I think we do have between the tissues and the capability to start to bring that together.
That population change with your there, because it didn't look like you're seeing a lot of your fully exhausted kind of go down, but I didn't see a lot of change in that progenitor exhaustion.
We didn't see it, but I think we always have to look, I think it depends on the tissue niche. So I didn't show that data, but we do have some evidence that the tissue niche probably matters. So far, we've been looking at very solid tumors in the marrow, but I think that we have other data to suggest that it depends on where the cells are. So I think.
Thank you so much. It was brilliant and truly enjoyable. And my question is it possible to establish the difference or the similarity in the signatures, in the genomic signatures, transcriptional signatures of activated T cells or activated post-exhausted state with the autoimmune signatures, T cell signatures? Is there any data there?
I haven't looked. There may be data there. There's a lot of data out there. I think we're at a really cool time because you generate a data set and then you realize that there's all this other stuff that's out there that can help you. And I think we should all be looking actively to see what existing data is out there. I'll give you one example. I think one of the early things that Erin did with her Richter's work is that, she was working in marrow. Turns out Chris Horrigan and the NCI created like this very nice resource of marrows, normal marrows that had just been generated. So useful. And then after we started to look at our populations to be able to look at data sets that have been curated by Ziming Zhang across different solid tumors, and then so I haven't looked. That doesn't mean that it's not there. And it doesn't mean, and if it's not here today, it might be here in 2 weeks. Or so I think it does behoove us to continuously go out there and look and see what's out there.
It's a cool question. I feel like vitiligo versus
Absolutely.
the incidence of vitiligo.
Absolutely. And I think that there's an increasing interest in immune-related adverse events. I think that there's going to be more and more activity in that area.
John?
Yeah, Catherine, great work. What about the immunophenotype of the exhausted cells? You talked about those early on, and you talked about checkpoint PD-1 blockade at the end, but was there as much diversity in the pattern of checkpoints and the phenotype of those exhausted cells as there were in many other parameters you looked at?
That's not jumping prominently to my mind. I'm sure, we did have site-seq data as well. And so that's not prominently coming to my mind.
You mentioned TIGIT as one, but it wasn't clear if those in those melanoma samples, if some of those, what was the exhausted phenotype that was?
Yeah. So I think,
Shoulder.
yeah, it's not coming to my mind. But it could be that I'm just confusing postdoc project 1 and 2 and 3. So, yeah.
Questions online there. Did you compare the T cell repertoire between the chemo radiation treated head and neck with the anti-PD-1 and the adjuvant therapy? We want to see if there's a difference between the immune cells compared to the standard therapy. HPV positive.
Yeah, we didn't. This was in the HPV negative setting. And Ravi promises me that we have a lot of work to do.
And the other one is, how do you comment on the recent Moderna progress on cancer vaccine? Do you think will be the future of cancer vaccine?
No one knows. I mean, again, I think, vaccines work differently than other therapies. They're a little bit more of a subtle therapy. I think there's going to be optimal settings.
The vaccine is, yes.
I do not know. I have no information.
Sounds about.
Yeah. So I think,
like
so I think it's going to roll out over time, but I do think vaccines have a valuable place in early disease, after therapy for kind of in settings of minimal residual disease, maybe recurrence prevention. I think the usual, which is to take our sickest and most treated, that's not a great place for a vaccine. And that's where a lot of therapeutics start. But it's trying, the vaccines ask something of the host, which is you got to mount an immune response. So you just have to keep that in mind. Yeah.
I was really intrigued by the CXCL10 story, and I know you didn't have too much time to get into it, but we hear a lot about T cells, whether it's adoptive T cell therapy or endogenous T cells. And interferon signaling is involved in various stages. And some people say it's good and some people say it's bad. So you've looked very specifically, you've got transcript data on a lot of these different situations. And what's going on here? You needed some interferon to get CXCL10 upregulated.
I think you have to design the right experiment to answer that question. I think we could do a phone conference with Marcella Mouse and see what she has to say about all of that. So I think that it is the right time and place to actually ask those questions, because with the right tissues and the right baits and targets, I think you could do a systematic look at all of those cytokines and try to maybe sort out in time resolved fashion the cascades.
Asking about the importance of bringing in T cells or.
Yeah. There was a lot of T cells there, and you showed in your one of your first slides, most of them aren't that helpful. There's only a few, and so are the others just coming along because are the Tregs making chemokines?
Exactly. So I think
effective futile
right.
recruitment.
And that's why I talk about the archaeology aspect. There was activity.
Right.
We're catching them at a time where the productivity was not good, right? The tumor was baseline growing, and yet there were all these T cells in the grave, so to speak, not all of them in the right, with the right specificity. The ones that did were pooped out. So I think it's actually the beginning of trying to sort out the chronology of events.
Super important, obviously. All right, one last question. We got to go.
Okay, so yeah, thank you very much. It was remarkable insights on this neoantigen based vaccine therapy for immunotherapy, basically. So if you would like to give comments on comparison of CAR T cell therapy, chimeric antigen receptor T cell therapy, and also aided with mRNA based vaccine, and the combination therapy of CAR T cell and the RNA based vaccine. There is recently the studies are there for solid tumors because they work well with hematological tumors, but not with solid tumors. So if you will compare this CAR T cell therapy and CAR T cell therapy plus mRNA vaccine with your new antigen vaccine therapy, what would be the better option according to with respect to the patient compliance and the outcomes in patient with therapeutic outcome, as well as the cost effectiveness? What would be your comments on this?
So there's about 5 questions in there.
Sorry for that.
So I can't comment too much on CAR T cells and vaccines, only to say that I am a big proponent of combination therapy in general. And you have to understand that kind of my roots are in stem cell transplant, which I consider the ultimate combination therapy, where you have kind of multimodal mechanisms trying to hit this very difficult problem of cancer in general. So if you have a way to synthetically get the right T cells to where you need to, great. If you have a vaccine that can bring new T cells and kind of drive a longer view of immune surveillance and immunoprevention, great. So I think they're kind of hitting different aspects of the problem. Right now, everything costs a ton. And that's why I do say that, if we are seeing positive signals and we are going to get to a place like, how sustainable is it to have these kind of high price tag therapies like CAR T in the long run? I think right now we have a very bad problem. Patients have cancer. We have to treat them, but kind of, is there a way to sustain all of this with lower cost, shorter time, et cetera, et cetera?